This is raw FASTQ data. Every minute, modern next-generation sequencing hardware generates millions of lines of unstructured text exactly like this. To process this volume, bioinformaticians rely on automated algorithmic pipelines and statistical variant callers to sort, align, and filter the data. However, these statistical tools often operate as a black box. Without a way to see the underlying reads, it is difficult to determine if a result is a genuine biological variant or a computational artifact. Relying solely on statistical abstraction is risky when interpreting complex structural anomalies. At this scale, even a small error rate in the algorithm can lead to thousands of false positives. Human visual verification provides a way to cross-examine these algorithmic results. By inspecting the raw data directly, researchers can determine which statistical calls are supported by physical evidence. The Integrative Genomics Viewer, or IGV, was developed by the Broad Institute to address this need for visual data exploration. IGV serves as a spatial integration engine. It allows researchers to overlay various data formats, such as BAM, VCF, and BED files, onto a single unified interface. The platform is interactive, enabling users to zoom from a macroscopic, chromosome-level view down to the single nucleotide base level. This interactivity is maintained even with massive, high-throughput outputs from modern hardware like the NextSeq 550, generating more data than traditional viewers can process. By aligning these different file types into a single coordinate system, IGV provides a navigable map for genomic discovery. The IGV workspace begins as a blank Cartesian coordinate system that must be constructed layer by layer. The first step is establishing the baseline x-axis by selecting a reference genome. This reference is typically derived from FASTA files. It is critical to verify the genome build compatibility here. If the reference genome does not match the sample data, every subsequent layer will misalign. Once the axis is set, researchers add structural information using BED and GFF or GTF file formats. These formats contain the coordinates for known genomic features, including the locations of genes, transcripts, and regulatory elements. IGV parses the coordinates from a GGF file and maps them as navigable visual geometry. These blue blocks represent known exons, providing context for the raw reads. These annotation tracks provide the necessary context to interpret the raw sample data that follows. After setting the foundation, the actual biological sample reads are mapped against the reference genome. This aligned sequence data is typically stored and loaded from BAM or SAM files. IGV displays these files as stacked read alignments. Each gray block represents a single sequenced fragment of DNA, positioned according to its alignment with the reference. To analyze this data at scale, the software generates a coverage track summarizing the depth of these stacked reads. This line graph tracks the density below. A localized drop can indicate sequencing biases or a physical deletion in the DNA. Inspecting the physical patterns in this stack, such as soft-clipped edges or unusual gaps, allows researchers to identify structural variations that automated callers may overlook. Visualizing the BAM layer maps the mathematical output of the aligner into a spatial representation of the physical DNA. We then shift from broad coverage to specific anomalies flagged by variant calling algorithms. These proposed variations are logged in the VCF, or variant call format layer. IGV displays single nucleotide polymorphisms and deletions on this dedicated VCF track, marking their exact position relative to the reads and annotations. A standard practice is cross-referencing every VCF call with raw read evidence in the BAM track. This allows researchers to visually confirm if a variant call is genuine or triggered by poor alignment in the underlying reads. This verification process provides the final check on algorithmic results, using the physical geometry of the reads to confirm the variant. The coordinate system used by IGV is not limited to DNA sequencing. It accommodates a variety of biological data types. Continuous data, such as signal intensity or expression levels, is visualized using WIG or BigWig file formats. In RNA-Seq analysis, these formats allow IGV to display gene expression levels and splicing information as continuous area charts mapped across the gene architecture. Researchers use the same tools in epigenomics to visualize DNA methylation or histone modifications across specific genomic regions. Because these different modalities share the same x-axis, IGV can stack multiple datasets vertically for comparative analysis. This visual comparison allows researchers to see differences instantly, such as a high expression peak in a tumor sample that is completely absent in a normal tissue control. The utility of this system lies in multimodal integration, allowing researchers to correlate DNA mutations, epigenetic changes, and RNA expression within a single window. A complete analysis in IGV involves selecting a reference, loading annotations, mapping alignments, and cross-referencing variant calls. The most important step is ensuring the genome build matches the sample data before loading any files. To ensure the research can be reproduced, it is necessary to document the specific reference versions and parameters used during the session. While visualization is a powerful diagnostic tool, visual patterns should not be interpreted in isolation. They must be supported by statistical evidence. By using visualization and statistical pipelines together, researchers can validate their findings and reduce the risk of false conclusions. As sequencing technology advances, platforms like IGV are evolving to manage increasingly large and complex datasets. Emerging trends, such as single-cell genomics, require tools that can visualize the diversity of individual cells, rather than the average of a bulk tissue sample. We are also seeing a shift toward cloud-based analysis and AI-assisted interpretation, where algorithms flag potential anomalies for human review within the viewer. In modern research, IGV functions as a primary integration node within high-performance computational biology pipelines. By aligning complex computational outputs into a navigable coordinate system, IGV enables researchers to resolve genomic patterns that remain hidden in raw data.